Welcome back to Sapio with Buck Joffrey. This is Buck Joffrey. Today we have a very interesting podcast. It is about nanoparticles through a company called Clene Nanomedicine. And we're going to have this really interesting discussion with Mark Mortenson and Rob Etherington, who are from Clene Nanomedicine. But before that, I felt like because of the complexity of the information here, I just wanted to kind of give you a little bit of an introduction to what this discussion is about. And then when you listen to it, hopefully you'll get more out of it because, again, it gets really into the weeds with some of the language that's used. But first of all, what are nanocrystals? Well, these are tiny particles. So small, in fact, that 10,000 of them could literally fit across the width of a human hair. And so what this company Clene's unique innovation is creating what are called clean surface nanocrystals. They're clean because traditional nanoparticles can potentially have, I guess, toxic residues, but these particles are residue free, making them safe and highly effective. Now, here's how they work, though. These nanocrystals target mitochondria. That's what they're doing. The power plants of your cells, of course, that's what the mitochondria are. And they work to improve energy production. Essentially, what they're doing is they're boosting ATP, which is the energy source, as you know, of your body. And also theoretically can help to reduce some of the harmful byproducts from the mitochondria called reactive oxidative species. Essentially, these nanocrystals, these tiny little, engineered things are used to act like a spark plug to optimize cellular function. Because again, if you've listened to this show or if you listen to a number of longevity shows, what you'll find is there is clearly a role in the decline of mitochondrial dysfunction in aging. So, again, think of the mitochondria as the engine, and these nanocrystals are potentially providing an external source of a spark plug for them. So Clene, this company, is focusing on diseases like ALS, multiple sclerosis, and Parkinson's disease. It's been in clinical trials. You'll hear a little bit about some of those clinical trials on there. But what's important is that, these treatments, they seem to be pretty promising, and they're also very safe. There's 700 patient years of testing, no serious adverse events. They don't seem to accumulate, even though you've got these like crystals, they don't seem to accumulate in the body or cause toxicity. So, what's interesting is that while Clene is, they're focused on neurodegenerative diseases. So, the Alzheimer's disease, Parkinson's disease, that kind of thing. There's lots of potential applications that go beyond that because mitochondrial health, as we've discussed now, is really central to longevity as well. So this technology could one day be used systematically to improve overall health and slow aging altogether. And remarkably, even though you think about these like nanoparticles, a lot of them are gold and stuff, they're not terribly expensive. Anyway, I just wanted to give you a little bit of that introduction before we got into the discussion. Hopefully it'll give you some, a little bit of background to help you understand the technical jargon. With that, here's my interview with Mark Mortenson and Rob Etherington.
In pursuit of health and wisdom, Sapio with Buck Joffrey.
Welcome back to Sapio with Buck Joffrey. And today my guests are Mark Mortenson and Rob Etherington. They are from Clene Nanomedicine. Mark is a co-founder and chief science officer. He's an accomplished scientist and engineer with multiple degrees and over 30 patents. He leads a groundbreaking work on nanocatalyst drugs aimed at neurorepair and remyelination, currently in FDA trials for ALS. And Rob is the CEO, and he brings a lot of expertise in life sciences and leadership, guiding the company as it pioneers this new class of drugs to try to tackle some of the toughest neurodegenerative diseases today. Welcome to the show, guys.
Thank you, Buck.
Happy to be here.
So I want to start out with this, just kind of giving a background on the concept of nanoparticles. It's funny. I trained at UC San Francisco, and one of my attendings, my professors, Dr. Ivan El-Sayed, and I think his dad was also in this space too at one point, was studying nanoparticles, and it was like this was about 10, 15, maybe 15 years ago, and it seemed like so space age, completely out there and very interesting. But I suspect I'm probably one of the few people out in this audience that really has even heard of the concept of nanoparticles. So why don't we start with that?
Mark, please. Mark's the founder, inventor of this asset.
So my wife is a medical doctor, and she's former research as well as faculty at Ohio State. And my background is material science and physics. And during a conversation with her one day about mitochondria and the electron transport chain that we got into for a variety of reasons, she told me some things about how electrons work their way down the electron transport chain and how that might work. And I asked her, how do you figure out what the energy level of the electrons are? She said, well, there's some information out there that you can look at, and you can see what levels they were at and what levels they are at, and they give off a spectra. And so I began to do that. And my background commercially prior to those conversations was I found myself in the DuPont organization in a variety of different technologies having nothing to do with biology, armor, tank armor, personnel body armor. I had a top secret special access clearance for a spy satellite. I was using materials, material science, and in that process of manufacturing a number of different products, catalysts are used. And the reason those two things come together is when I saw that electrons were going from higher energy levels to lower energy levels, I coupled that back with some of my knowledge of how catalysts worked in a variety of different situations. And there's a lot of metal particle catalysts out there, like in your catalytic converter that changes carbon monoxide into carbon dioxide. And if you look at what most of those reactions are, they are reactions at the electron level. So a number of covalent bonds get formed. And so I had a what if kind of moment. What if we could take the concept of solid state physics, material science, generate something and create electrons that might positively interface with the electronic energy levels of the electron transport chain? That was the beginning. That was the basis. There were a lot of hurdles to get there. And fortunately, not being trained medically or biologically, I didn't dismiss all of them as impossible, but rather kind of said, why not?
Yeah.
And so the journey began. And the quest first required, can we get a metal into a nanoparticle shape by a process that's not going to hurt, not going to cause harm? And it turned out we had to invent an entire process to do that. Backtrack just a moment. The nanoparticles that you're familiar with, Buck, they've all been made by chemical synthesis. And the results and sometimes the pride associated with chemical synthesis is there's stuff on the surface of the nanoparticles. That stuff is actually necessary in some cases to keep the nanoparticles in suspension. In other cases, just residue that has chemistry that you can't get rid of no matter how hard you try to wash it away with radiation or others. Or if you do wash it away, they're no longer stable. We recognized that catalysis is very much a surface phenomena. And all of the articles that had been written prior to our invention, there were toxic effects at 2 nanometers, not toxic at 3, toxic effects at 4, not toxic at 5. And no one had recognized that the toxicity was not an inherent property of the gold nanocrystal, but rather the residue left on the surface.
So
Likewise,
just to back up a little bit. So, I mean, just to get everybody caught up, we're really talking about delivery of various drugs or chemicals or whatever through the body using nanoparticles. So basically some kind of, whether it's gold or some kind of other, I guess, non-traditional medical approach to delivery, right? I mean, I just want to make sure everybody's kind of getting the big picture here as well as the...
Thanks. You stopped me at the exact right place. That's
Right.
where the entire world was when we started contemplating this.
Okay.
So, for example, MD Anderson was a very large leader in putting things onto the surface of gold nanocrystals to direct them to the cancer tumor and then apply light to cause a localized heating and removal of the tumor. That's where the world was. Our concept was an advancement on all of that. So we are not a delivery system. We are a therapeutic per se. So that's the exact right question to ask. And the reason that no one had ever contemplated this before, Buck, was that residue that was left on the surface was either toxic or blocked the efficacy of the electrons' ability to react with the system they were in.
So
So it's
the
a
the
very
old
large.
paradigm, just to be clear, the old paradigm, and I really haven't heard anything about that research, nor probably is the audience, but it failed because of this residue basically. So you had, again, conceptually, you delivered, say, some sort of therapeutic, or it could be part of the therapeutic. You bind, say, gold particles, and then you heat them up because they're attracted to a tumor, and you destroy the tumor. That's the concept. But you were running into toxicity with that, and that essentially killed that concept at that time. Is
No,
that
that's
right?
that's
Yeah.
75% there.
Okay.
Let me take the rest
Yeah.
25.
Yeah.
So that was one of the uses of the nanoparticles. There were other, they act as a drug carrier too. So they were
Hmm.
actually had an antibody or something put on the surface and they would carry something else. So they were targeted because they were always thought as being inert, completely safe. If you would have talked to the president of MD Anderson when I did, like in the 2008 timeframe, he would knock on wood and say, yeah, gold nanoparticles are completely inert. They're a great delivery
Yeah.
system or they're a great targeting system. And the only reason he said that is he'd never seen a clean surface gold nanocrystal. As soon as you have a clean surface, it's no longer inert. Those electrons are available to be interacting with the
What do you
local.
mean by clean surface? Just let's just make sure we understand again. What when you say clean surface, what do you mean by that?
Lack of chemical residue that's artificially placed on there or inherently placed on there because of the chemical reduction processes
Okay.
from which they're made.
Okay. So so tell us a little bit about this clean surface nanocrystals and the specific role they play in cellular metabolic processes.
So again, fortunately, not being a medical doctor or a biologist, I made the assumption that when you this is an oral medication for all of our drugs right now. So this is not injectable; it's oral. Tastes just like water. You can't tell the difference between water and it. And when you swallow it, I recognize my wife helped me kind of go through the processes. It gets through your stomach wall, gets into the blood system. And then again, being a material scientist and having made lots of products, I know there's two things, thermodynamics and kinetics. Thermodynamics, what should happen in the perfect world, kinetics, what really does happen. Okay. So the kinetic side of this, I thought probably the most abundant protein in the body, serum albumin, might become the primary coating. I knew there were others. I knew there would be others there, but that was the primary guess. And then I recognized serum albumin was a carrier for a lot of different things in the body and thought, okay, our biological identity, our protein corona is probably serum albumin. All right. That ought to get me moving around in the body and not getting trapped by the systems that would kick you out. And it turned out that was a reasonably correct assumption. Incomplete, but reasonably correct. Subsequent work, we've determined that there's some other haploproteins that go on there that really assist in getting across the blood-brain barrier. Pardon, but a little bit of dumb luck. We didn't recognize what that corona would be, but we recognized if we could get a clean surface nanocrystal into the body and get an appropriate protein corona, we might go to those places of sites of inflammation. We might at least get to those places. So those were the original assumptions. Absolutely incomplete, and we've been filling in the blanks ever since we've done the first work. But that was the original concept. That's how it all started.
So when you're talking about the albumin and using potentially the albumin as a carrier, well, I mean, what's the role of the metal at all in this role? If you're basically using this the albumin as a potential, you're basically able to manipulate maybe the albumin, put something on there that you want it to get somewhere. What is the role of the actual metal?
So just like in a catalytic converter, the platinum
Uh-huh.
nanoparticles that are embedded in the cordierite clay body, when it sees the exhaust, its job is to take the CO that it's at its surface. And so CO is right here. And then get an O right there as well, bring the two together. That's what catalysts do, and off goes CO2, not harmful. In the body, my original concept was if I could get these if I could get these gold nanocrystals near the mitochondria, and we've now shown not only we get near, but we can get inside, that these nanocrystals are designed because of gold itself and because of the highly faceted, like a woman's diamond ring, very high faceted material at the nanoscale, 13 nanometers, which calibrates your listeners. 10,000 of those would be the diameter of one human hair. So they're really small. Once those things get into the local vicinity, why couldn't they act like a catalyst like every other solid state physics reaction I'd seen? Why couldn't they help provide electrons to that electron transport chain and just kind of lube the mechanism, help it go along? In other words, why not have it act like a proton pump?
So the target is essentially the mitochondria. Try to optimize the function of the mitochondria. Is that right?
Well, as we have distressed mito, it is correct. But as we have distressed mito from disease state or just aging, they don't work so well anymore. So their ability to get the protons pumped out through the various complexes, find their way to complex 5, come back down in, and have the rotor on the bottom mixed together with ADP, adenine diphosphate, and free phosphorus to get your ATP. The premise was if we could lube that whole system and make it go better, if it's being stressed, then we might be getting more protons outside the inner membrane and help then have those protons come back through and cause more ATP production. That was one of the early mechanisms that we hypothesized we can have an effect on.
And Buck, back to your note that most people probably don't know what we're talking about. Let me just use a visual aid. So this is the CNMAU8. This is 60 mils. So that's about between a quarter and a third of a cup. Patients are drinking it every morning in the clinical studies. As Mark said, if I was blindfolded and drank my glass of water versus this acid, I would not be able to tell the difference. In all of our clinical studies that we've concluded to date, numbering 7, 6 finished, 1 in process, we use purple-colored water as our placebo, literally purple-colored water. So that just is an example of what we're talking about when we say that the acid is easy to take. If I can drink a cup of water, I can drink this acid. If I have a compromise in my disease like ALS and I can't swallow food or water, well, we can give it in a feeding tube. In other words, administration is remarkably easy. And then another way to think about what Mark's explaining is the mitochondria is remarkably energy dependent for the ways we move and walk and talk and eat and chew and breathe. Everything that you and I take for granted that gives us life and may be compromised in a disease like ALS or even in different ways, multiple sclerosis. They have challenges moving, sometimes thinking, sometimes seeing. We're basically arguing here that our asset serves as a catalyst. A nanoparticle is driving into the mitochondria, crossing the blood-brain barrier to enable the energy metabolic support that the mito needs to do its housekeeping and its job.
But ultimately, again, we're talking about, just improving the function of the mitochondria. I mean, that's the main goal here.
That was the premise of how we started. Yeah. There's some other glycolytic reactions that benefit as well. So less reactive oxygen species, more ATP.
So let's talk about specific applications because you're using this in neurorepair. The idea is that it can accelerate neurorepair, neuroprotection, remyelination. What are the mechanisms of this? Is it, beyond better mitochondrial function, are there other mechanisms that you purport that cause this?
So Mark, maybe will you take mechanism first, Mark, and then I'll take the clinical work and the focus there.
So we keep uncovering, Buck, a number of additional benefits. So, ATP is not just energy, it's signaling molecules. One of the first things that we did that will maybe help your listeners is when people said, how do you know your gold is really catalyzing anything that matters? We put human NADH molecules into a test tube. And we added gold nanocrystals to that test tube in varying concentrations. And we watched by visual spectroscopy the change in the peak that corresponded to the amount of initial NADH to 0, and the peak corresponding to NAD plus go much higher. So as a function of concentration, we showed that in water, our gold nanocrystals changed NADH into NAD plus. That's a very important thing. That's very important in our energy pathways. And so we thought as a mechanism, that helps to explain just by like the catalytic converter. Here's the incoming material, NADH. It finds the surface of the gold nanocrystals. Electrons are exchanged. There's a reduction reaction, and you end up with NAD plus.
Got it. Okay, so that obviously the NADH the NAD pathway is across the board in longevity something that's been studied quite a bit, even just in terms of aging across the board. I guess one of the advantages you're talking about here is an increase in the amount of that chemical inside the brain. Can you not get that from the NAD precursors that are out there?
No, you can't. So it's equivalent to trying to put more fuel into the engine, where we're in tuning the engine up. We're giving it new spark plugs, better oil, where you're just trying to get premium instead of regular in. So the two together, that doesn't hurt. But no, our mechanism is completely different. We are inside the cells, helping the normally occurring mechanisms just go a little bit better. And now, if they're not compromised at all, nothing happens.
One of the things about mitochondria that are tricky, and talking to a fairly respected name in longevity, Aubrey de Grey, and Aubrey describes the part of the paradigm of the aging in humans is that the very things that we need to function are the very things that ultimately start to make us break down, and part of that is mitochondrial function. So if you are increasing the function through this catalytic reaction of those mitochondria, are you also potentially increasing the reactive oxidation species and creating potentially more damage from that as well?
Fair question. We've actually done all those measurements in vitro and in vivo, and the answer is no. So a catalyst in a local environment really seeks homeostasis, seeks a balance. And once the balance is achieved, it's got nothing else to do in that local environment. So effectively it stops. So our experiments have universally shown, whether in a test tube or whether in animals when we're measuring, that we have less reactive oxygen species and more NAD plus. And we've got publications in Nature that go to those exact points.
And a follow-up question to that would be, if indeed the machine that this mitochondrial machine ultimately is part of our the damage from the reactive oxygen species are adding to our aging process, is there a way to use nanoparticles to specifically target reactive oxidative species within mitochondria?
I'd say there is, Buck. Let me tell you the one animal model that we did that maybe helps ground a little bit here. So there was the very first animal model we did was the cuprazone animal model. We did it in-house, and we subsequently did two more outside in much larger numbers, looking at both how the brain was changed as well as kinematic function. We had a direct correlation between brain changes and kinematic function. The cuprazone model that we did actually buggered up complex 1 and or complex 3, depending which paper you read. And so there was a buildup of reactive oxygen species, less ATP and more reactive oxygen species. We were able to show after just a 3-week intervention, after 2 weeks of poisoning with cuprazone, followed on by 3 more weeks of poisoning with cuprazone, that we'd brought back not only the cellular structure, the neurons with remyelination, but also it brought back motor function, the ability of in this case, the mice to run around. We did a rat model as well, where we compromised all of the neuron mass, and in a matter of 2 weeks, brought it back. This is all about energy production and the highly energetic needs of the brain.
As a physician, I'm also curious about the role of inflammation in neurodegeneration and aging. How do the nanocatalysts interact with inflammatory pathways, and is there potential for them to modulate chronic inflammation in the neurodegenerative context?
Rob won't want me to speculate too far, but that's actually some of the very first things that we tried and confirmed. And so the reason I mentioned serum albumin, wherever you have sites of inflammation, those sites of inflammation love serum albumin because they've gone from the regular Krebs cycle to the Cori cycle. And so they start eating away. And so serum albumin, I used to use the metaphor, it's like cheeseburgers for hungry teenagers. They just go there. And so once we get there, are there inflammatory moderators? Yeah, there are. But we've left that pathway for the moment. It doesn't mean we won't come back to it.
Talk a little bit about your your challenges that you've encountered bringing some of the nanocatalyst drugs to market? I mean, obviously, this technology is relatively new to the FDA.
So why don't let's tackle that in two parts. Mark, why don't you talk first about manufacturing, our IP and our trade secret piece, because that's a key commercialization element, and then let me jump into the clinical piece.
I'll give you the quick 30 seconds. So in order to get a clean surface nanocrystal, you can't go anywhere and buy it. They don't exist anywhere in the world. So we had to invent a way to do that. We've built a very robust process now where we can make gallons an hour out of each of our devices. The devices had to be invented, the process had to be invented, and it's a novel material. No one has ever before made a clean surface gold nanocrystal without things on the surface that are suspended in water. So we were the first to do that. So there's IP on the method of making the material, the device in which it's made, as well as the material per se, as well as method of use. So it's a kind of a clean sweep on the patent side, and we can do it to GMP conditions now. The clean rooms are right behind where I sit.
And we've completed clinically 2 phase 2s in ALS, the Healy ALS platform study, which was done with Harvard, Mass General, and 57 other clinical sites of excellence in the ALS sphere across the United States. That was a 6-month study, very novel approach. And though we missed a primary endpoint in that program, so that speaks to one of your challenge questions a second ago. We missed the primary endpoint of ALS-FRS, which is a classical endpoint used in ALS studies, but unfortunately missed by 9 different drugs in the last 18 months alone, including Clene. We also did a 9-month program of a phase 2 in Australia with the 2 top ALS experts there. We've completed a visionary study, we call it, in multiple sclerosis. And in that case, we were simply trying to see if we could take patients with optic neuritis, so a compromise of their visual field because of multiple sclerotic lesions on their optic nerves, and we could help them see again. That was the primary endpoint. And the secondary endpoint was to see if we could help them think again or move more effectively again. And we've already released this data in this phase 2 proof of concept study and showed that we did help them see again, actually, and also helped cognitive improvement as well as the multiple sclerosis functional composite scale, which is an ability to see, move, walk, and think. And then we've also completed a study in Parkinson's patients that was an early POC to show target engagement. The drug taken by mouth gets to the brain and does what we hope it's supposed to do, which it did do and was statistically significant. That is improve nicotinamide adenine dinucleotide. So this metabolic effect that we're speaking to needs to occur to give the mito the energy it needs to fight back against the Parkinsonian assault. And we have ongoing right now a study in progressive MS, which is a true market need. Patients who are progressing in their multiple sclerosis despite being fully controlled with their DMT. So a lot of different neuro approaches in 3 different areas: ALS, Parkinson's, and multiple sclerosis, all of which we've tackled at the same time. And as we've also announced publicly, we have ongoing regulatory conversation with the US FDA to consider whether or not they would let us file an accelerated approval application on the basis of a surrogate endpoint neurofilament light, which is a biomarker that Clene has seen statistically significant data in our Healy program, and which the agency earlier approved Biogen's tofersen for SOD1 ALS patients on neurofilament light alone. So we are presenting our survival conversation and our neurofilament data to the agency, and we'll find out their thoughts in the coming months.
Have you noticed any, I mean, what if any are some of the major risks of this kind of intervention? And is there anything related to the actual particles themselves? I mean, how are those eliminated? Okay. Is there
So let me give you the headline there and then let Mark talk about some of the elimination side. So on the headline, we have over 700 years of subject exposure without a single identified safety signal against ALS, multiple sclerosis, or PD. That's a pretty audacious statement to say, but the safety aspect of CNM-Au8 has not been in dispute. In fact, the safety conversation was addressed early on with the FDA. When I say early on, I mean 6 years ago, because they had the same elimination questions that you just had of an asset taken orally and is excreted. But we have not a single serious adverse event, not one single serious adverse event that has been related to CNM-Au8 in any of our 3 disease programs to date. We do get some side effects. Patients sometimes complain about nausea, headache, diarrhea. And so those we see different, but those are mild to moderate, transient, and have not in any categorical way been showstoppers. We also have no identified drug-drug interactions, but it's because of the way our drug particularly is metabolized, which is to say it's not really metabolized at all. Mark, do you want to comment?
So the gold nanocrystals are primarily excreted through the feces. They come out exactly the same way they went in. So as opposed to any other drug you've ever seen, Buck, there's no metabolism changes. You do not digest gold nanoparticles in your body. So the highly faceted gold nanocrystals come out just as they went in. If you were to use any other gold nanocrystal made by any other technique in an ingestible way, you would have potential very strong safety concerns depending on what that residue is, that not clean surface. So there might be zero efficacy and there might be toxicity. You won't have that with a clean surface though.
All the disease processes we've been talking about all relate to the brain. Is there any reason for that specific target? I mean, obviously, I don't know if we've necessarily touched on it, but there's some unique qualities about this that allows for penetration through the blood-brain barrier. But is there any reason to believe you couldn't use this for any and all type of rejuvenation? I mean, certainly just in longevity in general, mitochondrial health is a huge thing, right? So I'm just curious, is there any reason to think that this wouldn't be beneficial systemically rather than just in the brain?
I think systemically it's a wonderful approach. And I believe that there's lots of opportunities to optimize what we've done. What I sometimes proudly say, as opposed to having iterations back and forth with a manufacturing technique, the output of the manufacturing technique in biology, we had mathematical calculations and we locked in our composition without a single piece of biological data. And there was reasons that we did that. And so, to tell people biology is math is a little bit off the charts. To tell people that we locked in our composition without a single piece is a little bit off the charts. And so if we had additional resources and time, we could fine tune these gold nanocrystals and maybe others. Our platform technology, we can make things nature can't make. So we can make mixtures of materials into a single nanocrystal. We can make composites, a little bit of something on the surface of another. We've proven to ourselves we can do that. And the reason that being important is there's different electronic energy levels that are available by these different techniques. So to your point, Mark, could you make a different nanocrystal, put it somewhere and control a cancer metabolism, or speed something up or slow something down? Catalysts can do both, right? So in the world of solid state physics where I come from, if you want to slow something down, there's a catalyst for that. If you want to speed something up, there's a catalyst for that. So all that remains untapped. We have the platform to do it, and we just need the wherewithal of resources to get there.
How expensive is this stuff? It must be, I mean, if you're dealing with gold particles, that kind of thing, it might get a little pricey.
It's not, because while Rob will tell you there's quadrillions of them in a few ounces, as far as volumetric, it's not much. So, our drug is 99 point something % water. So the amount of gold that's actually in there, even though it's a color because the gold is there, is very small. And so, it is a very affordable and very robust process. And it can provide, I think, universal applicability across the neurodegenerative space, the whole neurodegenerative space, and can't wait to get there and prove that to be true.
And adding some of your context, we typically explain this. We're more expensive than a small molecule, which is just a recipe of chemistry stirred in a vat. But we're a lot less. So we're more expensive than that, but we're a lot less expensive than a classical biologic. These inputs that Mark just spoke to, water is one, gold is the other, electricity is another. Not just any electricity, not just any water, but still, the costs can be manageable.
Right.
And that's at present scale. When we actually get to what we expect to be C's volume scale as Clene matures, then those costs will drop.
Yeah. Great. Well, guys, thanks so much for your time today. And how do people learn more if they want to learn about what you're doing?
I think they should go to clene.com. There's also a pretty interesting mechanism video there and some other description of our clinical studies, www.clene, C-L-E-N-E, and it's pretty expressive. And they can also reach out to us through various links on that same website if they so choose.
Thanks so much for joining me, and it's been a really interesting conversation.
Thanks for having us,
Good luck
Buck.
to you.
Thank you, Buck.
Thanks for listening to Sapio with Buck Joffrey. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe. Until next time, this is Buck Joffrey for Sapio with Buck Joffrey.